So, good evening, everybody. Today in the introductory sessions that we are taking, introduction to microbiology, especially for the newly joined first year PGs in mind. Today, we have Dr. Sunita Sahu, who is the senior consultant and head at Apollo Hospital, Bhubaneswar. And she's going to talk about microscopy, basics, and advance, and how microscopy can assist diagnostic uh, capabilities and give us a skill which is very, very important. So, over to you, Dr. Sunita.
Thank you so much, ma'am. And good evening, everybody, and good evening, ma'am. And to start without wasting any time, the topic given to me is microscopy, the basics and advanced. And let me start. Okay. So, today evening, while I was coming to the department, I got a call from the radiologist. He had rung me up and told that this patient, who is 56-year-old diabetic man, has multiple hepatic abscesses with enhancement, and he has underwent a CT guided liver abscess aspiration. They are in a dilemma because the patient was on meropenem for quite a long time and was being treated as per management protocol. So, as a clinical microbiologist, when I have to get that sample, I really have to help the clinicians and also the radiologist because he had called me and told, Madam, we are clueless, which organism will be there, and because the patient is not responding to the carbapenems and patient is deteriorating with the abscess being increasing in size. So here the role of a clinical microbiologist starts, and when the sample comes to the laboratory in the microbiology department, we do a direct examination microscopically and also macroscopically, and we grow it and cultivate it. And if required, we do the nucleic acid detection. Nowadays, we do quite a lot of nucleic acid detection also. So, having said that, the two important qualities of a microbiology lab is a shortest TAT and a high accuracy. Shorter the time we take to give the report, it is better. But at the same time, the report should have a high sensitivity and should also be specific in what we give. Now, keeping the patient in the back of my mind when I will receive the sample, I have to evaluate the specimen quality. I also have to see whether the organism is cultivable, and if it is not cultivable, how fast I can give the report to the clinician and the radiologist so that they come to a diagnosis, and do a direct microscopy to evaluate in order to correlate whenever I get a colonial growth in order to compare with the growth characteristics. So, little bit about the history of the microscope is that it started from the 16th century when Janssen invented a compound microscope having two lenses. It was later a little bit, it was advanced by the scientist Robert Hooke, who designed a two lens microscope and with a better clarity, followed by the Dutch scientist Leeuwenhoek in 1670 when he developed the microscope with a single lens, but the clarity was not much. Then in the 19th century, that is in around mid 19th century, scientists Abbe and Carl Zeiss, they brought up a good lens system, good microscopy system in which we got a better clarity. So this is how the microscopes have evolved from a tube-like structure to having binoculars and having better lens clarity. And we can see with a better clarity and give proper diagnosis and a early diagnosis to the clinicians. Now, the microscopy applied in the laboratory diagnosis of microbiology are many because first is it gives a rapid preliminary organism identification because we can directly visualize in the patient specimens. Sometimes we also can give a rapid final identification of certain organisms because we are directly visualizing it. We also can detect different organisms. Suppose there is gram positive, gram negative, or any nocardia type species, we can also identify that immediately. Now, we also can detect organisms some organisms which are not easily cultivable, like Haemophilus influenzae, and sometimes like Brucella, sometimes they are not being cultivated. So, those times it becomes easier for us to see the gram stain and give the report. We also evaluate the patient specimen, because I told that when I will receive the sample, I will evaluate the specimen for the presence of the inflammatory cells, for the presence of squamous epithelial cells or any liver cells present. I can determine the organism's clinical significance, because there are certain organisms which are prevalent in liver abscess. So if it is correlated, I can determine and give a quick diagnosis to the clinicians. Pre-culture information is very important, because sometimes it happens that we do not get any history from the patient, from the clinician side or from the radiologist side. But this time we were lucky that we got the information and we know that what organism may come and how to cultivate it. And after we know the organism which is present in the gram stain, it will be easier for us to cultivate. If it is a acid fast bacilli, or if it is a nocardia, or if it is any non-culturable organism which is present in the gram stain, we can go for nucleic acid detection. Now, similarly, to elaborate more on use of microscopy, it magnifies the objects which are too small to be visualized by naked eye, and it directly detects the microorganisms from the clinical specimen. We can see the multiple morphotypes or single most prominent pathogen which is present. We can give a preliminary detection of organisms and that are not easily cultivable. We can evaluate the specimen quality, and direct microscopy evaluation is used to correlate with colonial and growth characteristics. Once we get tomorrow the growth characteristics, the growth of the organisms can be compared with the gram stain which was done today. Now, indirect microscopic evaluation of the pure colonial growth can also be correlated. So these are the uses of microscopy which is to be used in the laboratory in order to give the diagnosis to the clinicians. Now, coming to bright field microscopy, which is very usually used, it happens that the visible light passes through the specimen and through the lens that results in the magnification of the organisms. There are three main principles to it. Objective lens of 10x, 20x, 40x, and 100x, and it varies with the microscopes that is present in the laboratory. The ocular lens magnification is 10x. Then comes the resolution. In resolution, there are details of the magnified object. The resolution maintains the details of the magnified object. It is defined as the closest distance between two objects that permits definition of each. Meaning we can come to know whether those two objects which are very closely placed, whether they are different objects or whether that is the same object. So that is what is meant by resolution, and it is the second principle of the bright field microscopy. And the resolving power is the characteristic of the microscopes, and it is based on the manufacturer's classification. Immersion oil is used to increase the resolution based on the type of microscope which we use. Then the third principle is the contrast. Now this principle helps in differentiating the different type of organisms which are present in the gram smear. So these are the three main principles, that is magnification, resolution, and contrast, which is used in the bright field microscopy. Now, a little bit elaborating the bright field microscope. This is the ocular lens. Then the oil immersion objective lens specimen on the glass side is present. We put the immersion oil, that is the stage. The condenser is present below, and this is the light source. So this whole thing is present in the bright field microscope, which we use in the laboratory quite often. Now, this is a diagram, schematic diagram. This is where we see the ocular lens, which is close to the eye, the objective lens, which is close to the specimen, and then the condenser, which is below, and the light comes here. This is the light path which goes through the condenser, specimen, objective lens, ocular lens, and which goes to our eyes and we see. And in our eyes, we see the magnified image, and this is the magnification. That is why we use the bright field microscopy so that we can see the organisms or see the objects which are not usually visible in naked eye. Now, covalent illumination is used because it provides the maximum illumination and resolution, and this is to be known to us who are using the bright field microscopes, because if the condenser is not positioned properly, we will not get the covalent illumination, and that is why this has to be known by one and all who use the light microscopes. Now, the features of the light microscopes is, as I detailed before, is magnification. So lens eyepiece magnification is around 10x. Now the resolution is equal to the ability to differentiate 2 points as separate, as I described before. Now, naked eye will see around zero point 0.2 millimeter. Light microscopy will have a resolution of zero point 0.2 micron meter, and electron microscope will have a resolution of zero point 0.2 nanometer to zero point 0.5 nanometer. Now, this characteristic of features of light microscope is important in order to understand where we can use the light microscope, where we can use electron microscope, in order to give the diagnosis to the clinicians after receiving the samples. Now, the third thing is the contrast. Now, this contrast helps us in differentiating the different types of organisms in the light microscope, and this contrast in light microscope is got with the help of the dyes, and which will be elaborating in next few slides. Now, the objective lens, we have scanner, low power, we have high power and oil immersion. Scanner has got a power of 4x, low power 10x, high power 40x, and oil immersion is 100x. Now, eyepiece lens is fixed, that is 10x. So, when we calculate the total magnification for the objective lens scanner, which is 4x, when we multiply with eyepiece 10x, the magnification becomes 40x. For low power, 10x magnification when multiplied with the 10x of the eyepiece lens, the total magnification comes to around 100 times. The high power objective lens has got a 40x magnification when multiplied with 10x eyepiece, gets a total magnification of 400 times. Now, oil immersion has got a magnification of 100x. When multiplied with eyepiece magnification of 10x, we'll get around 1000 times total magnification. So this is how we calculate the total magnification. Now, to calculate the total viewing magnification of an object when we use a 40x objective lens, this I have posed as a small question for the students who are attending today. Can anybody calculate if we use a 40x objective lens, what will be the total magnification? Those people who are attending can write it in the chat box. Sorry. Okay. So to calculate it, since it is a 40x objective lens, that is high power, eyepiece is 10x, so total magnification will be 400 times. So little bit about the direct and indirect smears when we are talking about microscopy. Now primary clinical sample, when we make a smear, that is a direct smear, and the use of it is it identifies the type of cells present in the sample and the microorganisms, the white blood cells and epithelial cells. It is used to provide preliminary information to begin patient care and determine identification method. As I told you the history, today when I came to the department, the radiologist wanted to send the sample so that we can provide a preliminary information like what are the type of cells, if there is any organisms, what are the type of organisms present, so that they can start the patient care and determine the, and we can help in identifying using which type of media for the organisms. Now the other type of smear is from the cultivated microorganism. So once we cultivate it, if we make a smear, that is known as indirect smear, and it identifies the single morphotype of cells from the isolated colony, and it becomes mandatory on our part as a clinical microbiologist to correlate between the smear of the cultivated microorganism and the direct organism which is present in the direct smear. So that is what I told, that it is used to correlate with the initial direct smear for identification and resolving any discrepancy if noted. For example, if we see gram-positive organisms in the direct smear, but we do not grow gram-positive organisms, then we have to go back and check our direct smears and then correlate with the indirect smears and finally give a diagnosis to the clinicians. And that is why it is very important that we should know that when a direct smear is being done, it has to be monitored, it has to be told how it is to be done, from which part of the sample it is to be done, what sterility precautions have to be abided by when doing the direct smears. Okay. Now, a little bit about gram stain, since we are discussing about microscopy. The principle is a differential stain. As I told that the contrast is done with the help of the dye, and gram stain is an example where a contrast is used to identify the different type of bacteria. Now, the primary stain is the basic dye crystal violet. Mordant used is iodine. The decolorizer used is alcohol or acetone, preferably 1:1 solution, and the counter stain used is basic dye saffron. So, and these are the few pictures which I could get. Gram positives will stay, which stains deep purple, and gram negative, which will stain pink to red. Now, coming to little bit of rapid identification schemes, because as I told you, a very important quality of a microbiology lab is to give the report fast. So, how to give the report fast is we do gram stain. When we do a gram stain, we do see whether those are gram positive cocci, gram positive bacilli, or gram negative bacilli, or gram negative cocci. So I have done an algorithm. If it is a gram positive cocci, how to give a report fast is once it grows, we do a catalase, positive staph. If it is negative, strepto or enterococcus. With the gram positive bacilli, and we see for spores, if it is positive then bacillus species, if it is negative then catalase we do, and if it is positive catalase then corynebacterium species, listeria species or others. If it is negative then lactobacillus species and actinomyces species. But if it is gram negative and grows on McConkey, then Enterobacteriaceae or Pseudomonas, oxidase positive Pseudomonas species, negative Enterobacteriaceae. If does not grow on McConkey, but there are gram negative bacilli in the sample, we could think of Haemophilus brucella and Legionella. Now coming to gram negative cocci, if they grow on Thayer martin, then they are pathogenic Neisseria. If they do not, then they are other Neisseria species. Now, if this algorithm is present in the lab, it becomes easier for us when we do a microscopy and see for gram stain. And when we get these type of gram stain reports, we can correlate according to the algorithm and go ahead with our identification. Albert stain is for detection of volatile granules in diphtheria. And Albert, it has got two types of stain, Albert stain 1 and Albert stain 2. Albert stain 1 has got toluidine blue, which is a metachromatic stain, and malachite green and glacial acetic acid. Then Albert stain 2 has got iodine, and this is what we get at the end result. Now, impregnation stain is also very important because the stains gets deposited on the surface to make it thicker, and silver stains are used, and the thin structures like flagella and spirochetes are stained, and we can give a quick diagnosis to the clinicians like this. Now, acid fast stains is a differential stain which is used to detect bacteria whose cell wall contains mycolic acid, that is mycobacteria. Now, there are two methods, which is a Ziel-Nielsen stain. Primary stain enters into these stains cells by heating, and the Kinune stain, which is the primary stain, contains phenol, and it helps in fixing the bacteria onto the slides, and then it gets stained. Now, acid fast stains the primary carbon fusion. Because of the carbon fusion, it stains pink. The other object will stain the color of the counter stain, and the counter stain used here is methylene blue and green malachite green. So, this is how it looks, the acid fast bacilli, and this is a negative. Now, the microscopy in TB diagnosis or the National TB Elimination Program, because again, this is also a type of rapidly diagnosis. Now, it has got its own advantages and its own limitations. The advantages is that it is very cheap, it can be done fast, it has got a short turnaround time, it has got a high specificity, but it has at the same time a low sensitivity. It becomes positive only when there are more than 5,000 bacilli per ml, Hence, cases would be missed in early disease or when an inappropriate biological specimen is provided, where the bacterial load in sputum is less. Another limitation of microscopy for tuberculosis is it is unable to differentiate between Mycobacterium tuberculosis and non-tuberculous mycobacteria. Now, a little bit about the advantages and disadvantages of microscopy. As I told, it is rapid. Advantage of microscopy is rapid. It is low cost, non-invasive, specific and flexible technique. It provides physical features like color, homogeneity, shape, degree of aggregation. Multiple probes can be used, and data on membrane components of structure and dynamics can be obtained, and multicolor imaging can be reached with fluorescent microscopy, and obscure cellular subcellular images can be processed. Now, disadvantages is it is inadequate to produce conclusive observations and comprehensive information. Low magnification range from 500 times to 1500 times. Photo bleaching and loss of fluorescence intensity in case of fluorescent microscopy. And the properties of specimen can be affected by incorporation of the probes and the dyes. Now, the simple stains used in microscopy is methylene blue and basic fusion, which everybody should know about it. And I've just taken few photographs. These are Campylobacterium grams because now grams, if we do and if we rely on grams, then if we see such type of structures, sometimes it becomes difficult for it to grow. So that is why from grams then we can think of that probably this could be Campylobacter. And similarly, Brucella, it appears like grains on the gram stain, and this appears like this, and we can give a hint of the type of organisms present. Now, when we do a gram stain and we see it under the low power, as a microbiologist, we should see the WBCs, the epithelial cells, and larger organisms like fungi or parasites because budding yeast are usually seen uh, on a gram stain. Another important stain is the negative stain, and the stains used are Indian ink and nigrosin. And this is how it looks. Background is dark in color, and this is the capsule. This is the capsule. This is the bacterium. And a negative stain can also give us a hint. In case it is Cryptococcus, it can give us a hint about if it is present. We can tell the clinicians about it. Now, coming to the phase contrast microscopy, in the phase contrast microscopy, we have an annular ring below the condenser, which is absent in the light microscopes. And another is the phase plate, which is present near the objective lens. Now, what happens when the light source enters into the microscope? It crosses the annular ring and then it reaches the condenser. And from the condenser, the light is scattered and it goes again and hits the phase plate. And this phase plate gives the contrast, and therefore the organisms are visible bright because of the phase contrast created by the annular ring and the phase plate present in the phase contrast microscopes. Now, in phase contrast microscopy, we view the organisms and other cells in a wet preparation or a wet mount. Staining is not required, therefore live organisms can be visualized, which is advantageous. And beams of light pass through a specimen to create different light intensities and a greater contrast. So these are the way by which we can do a phase contrast microscopy, and we visualize the movement of the organisms, and therefore we can give a quick diagnosis and we can tell which type of organisms are present with the help of a phase contrast microscope. Now, uses is it identifies medically important fungi which is grown in culture, and it also has got parasitic identification. These are the extra uses of the phase contrast microscopy. Now, coming to fluorescent microscopy, now a little bit a schematic diagram of it. There is a light source, which then there is a excitation beam. The light gets excited. It hits this light wave of the splitting mirror. The excitation light then goes and hits the specimen, which contains the microorganisms which are stained with the fluorochrome. Then the fluorescent light passes through the passes through the objective lens and comes to the eye. So this is the principle on which fluorescent microscopy works. That is, the light source passes through the UV light, gets excited, the excitation light hits the specimen, which is stained with the fluorochrome, then it becomes a fluorescent light and it is visible to the eyes because it is differently colored. So the fluorochromes, the principle is in fluorescent microscopy, the fluorochromes are raised to high energy level after excitation. The excess energy is released in the form of visible light when the molecules return to the normal state, and then the objects appear brightly lit against a dark background. And this is how it looks based on the stains which are used as in the fluorescent microscope. Now the fluorescent dyes are the fluorochroming, the dye present is the fluorescent dye when it is mixed with the target bacteria or added to the target bacteria, they get the final end result is the bacteria stain and fluoresce, and that is how we get to see in the microscopes. Now, the immunofluorescence, the fluorescent dye is conjugated along with the specific antibody. The antigens are present in the microorganisms, and when the fluorescent dye with the specific antibody combines with the antigens, there is immunofluorescence, and that is visible in the in the microscope. Now the fluorochromes which are used is acridine orange stain, which stains the nucleic acid and is a non-specific cationic dye. It confirms the presence of bacteria in blood cultures when blood cultures are indeterminate. Calcofluor white stain binds with the cellulose and chitin. It greatly enhances fungal visibility in tissue and in other specimens. Now, oramin rhodamin stain is a basic dye that binds to the mycolic acid, and the cells appear bright yellow or orange against a green background. So these are the various fluorochromes which are used for fluorescent microscopy. Now coming to dark field microscopy, it is a the light is directed at an oblique angle, and that is how it achieves a contrast and it gives a better visibility. The only light that hits the object will be visualized against a dark background, and this is how a dark field microscopy. The light enters through a dark field and then the light gets scattered, and finally which we get is a brighter version of the specimen which is present in the slide. So the dark field is present, or the dark field ring is present by the side of the condenser lens, and the light which enters from the light source gets scattered, and then when we see it under the objective lens, it appears brighter against a dark background, and it is used to detect spirochetes, which is most notorious is Treponema pallidum, and it is a causative agent of syphilis. And this is how it looks against a dark background. These are very prominently visible if we use a dark field microscope. Now, I have one question here. The dark field microscopy may be used for the microscopic examination of these types of bacteria: gram-positive Cocci staph aureus, Yeast Candida tropicalis, gram-negative Escherichia coli, spirochetes I have already discussed. So it is an easy question. Students can write in the chat box their answers. Now coming to digital or virtual microscopy. Now, automation and digital imaging can be done. It has got the ability to adapt to microscope for multiple uses. Like the microscope can be changed into bright field with certain accessories. It can be changed into phase contrast and also into fluorescence microscopy. The slides can be scanned and interpreted with a web-based interface, and with the help of the software, the slide can be tracked. It can be standardized with proper quality control, and consultation can be done with transmission in real time of images and results. So this is how advanced it has become from the way back years of from the 17th or 18th century till now. We have moved ahead from a compound microscope to a better imaging, better resolution, and fluorescent microscope. Now we have come to a digital or virtual microscope where the slides can be put and can be scanned using a web-based interface. We can track the slides, we can standardize and do proper quality control, and we can also consult and we can also give our reports in a real time with the real time images. Now, these are the different morphology in gram stain. These are the gram positive bacilli without spores, and these are gram negative coco bacilli. These are few yeast cells present. So, this is the advantage of microscopy, that we come to know the cellular morphotypes, the microorganisms morphotypes, the amount of organisms present, the amount of cells present, and these all things will give us a idea about the samples, whether the sample is properly collected or not, or whether the sample has been contaminated while collection, and what is the pathogenic organism, which type of organism has got a is present in more amount, or which type of organism is present in lesser amount. So these all different types of information we can get with the help of the gram stain. So, some amount of the advanced, as I told you, that gone are the days where we were not able to share the reports which we used to see in the microscopes. We can use a web-based app and we can send it, and we can do consultation and we can ask our doubts if we have any doubts with the help of new advanced microscopes. Now, this digital holography microscopy has been used to visualize bacteria in aqueous environments without the loss of resolution in samples of 1 millimeter thick. This is approximately 100-fold greater resolution than most bright field microscopes. And although currently under development, this technique has the potential to improve the identification of low concentration of organisms in clinical samples. So sometimes it happens that in microbiology, when the amount of organisms present in the gram stain is very less, we are always in a dilemma whether to report or not. And sometimes what happens that when a gram stain is prepared from a sample, we do not get any type of organisms on the gram stain. So digital holography microscopy will help in those times where the concentration of organisms is less, to get a information about the organisms present in the sample. Now, the atomic force microscopy is another advanced microscopy. The advantage is that it can do a genetic, it can see the DNA, it can do a DNA study, and we can know which type of DNA is present. So that is the advantage. But the disadvantage is that it is costly and it is not widely available. Now, this is a chart for microscopy for diagnostic microbiology. Uh, for bacteria, bright field microscopy, digital microscopy, fluorescent and phase contrast, and dark field microscopy can be um, used. But when it is fungi, bright field, digital, fluorescent, and phase contrast can be used, but dark field microscopy cannot be used. Parasites, same, dark field cannot be used. For viruses, digital and fluorescent microscopy can be used, but cannot be visualized under bright field microscopy, phase contrast microscopy, and dark field microscopy. So these are the if we know these things, then when we get a sample with the clinical history, what is the clinical suspicion? If the consultant is having some type of suspicion, like whether it is bacteria, fungi, parasites, or viruses, we can take help of this chart and we can use the type of microscopy required. So in 2024, machine learning and AI, the advances in machine learning and AI are greatly reducing microscope data processing and image processing times, and in some cases from days or months to seconds. Because as I told, a very important part of a microbiology lab is to give a report faster at a shorter TAT, but at the same time with proper sensitivity and proper specificity. And that is how we can make a difference. That is how we can help the clinicians, and we can give a early diagnosis. And if we give a early diagnosis, that will help the clinicians in starting an early treatment to the patients. Now, the take home message is microscopy is an important examination, and specimens from patients suspected from fungal infection should be treated with considerable caution. Whenever possible, specimens should be taken from infected body sites. Specimens taken from infected or primary sterile body sites should undergo fungal microscopy along with culture and non-culture based methods. Swabs should be avoided and samples should be preferably collected before starting antibiotics, because many a times or most of the times we get a sample after starting antibiotics, and gram stain gets affected if the patient is already on antibiotics, because in literature it has been mentioned that if organisms have been exposed to antibiotics, their cell wall deteriorates, their cell wall gets destroyed, and if the cell wall is destroyed, it will not take up stains and it will not be visible. And even if it is visible, the gram positive may look like gram negative. So these are the few things which we should know, and we should be a guiding factor for the clinicians when they are sending samples to us. Now, that was my last slide, and that is why microscopy surveillance is crucial. And we should not ever think that microscopy is of no use. I feel microscopy is to be taken as a very important part of laboratory diagnosis in microbiology. Gram stain gives us lots of guidance, and gram stain should be done properly. All types of microscopy, all types of stain will give a rapid diagnosis. Let it be a Zn stain, let it be a Albert stain, or let it be a negative stain, or a wet mount. Any type of microscopy gives us a lot of clues and helps us in giving some diagnosis to the clinicians. I have few questions for the students. If they can give their answers in the chat box, I will just read out few questions. These are my references, and the questions are: the digital holographic microscopy allows for improved resolution over regular light microscopy by approximately 10-fold, 100-fold, 1,000-fold, or none of the above. You can write your answers in the chat box. The second question is: a fluorescent dye coupled with specific antibodies is called immunofluorescence, fluorochromic, actinic orange, or electron microscope. So that is the second question. I repeat it. A fluorescent dye coupled with specific antibodies is called immunofluorescence, fluorochromic, actinic orange, or electron microscopy. Please write the answer in the chat box. This third question is: the stain that binds to the nucleic acid of organisms but does not discriminate between gram positive or gram negative organisms is called Ziel-Nielsen stain, Oramin rhodamin stain, gram stain, acridine orange stain. I repeat the options: Ziel-Nielsen stain, Oramin rhodamin stain, gram stain, or acridine orange stain. These are true or false. First question is: the ocular lens is the lens closest to the specimen. Second question is: a fluorescent dye coupled with specific antibodies is called fluorochromic, whether it is true or false. Please write. You write like one, is it true or false? Two, is it true or false? Like that. Number third is: resolution is the extent to which detail in a magnified image is defined. Fourth is: fungal stain is a gram negative stain. Fifth is: acid-fast bacteria are resistant to decolorization. I will repeat the questions. The ocular lens is lens closest to specimen. A fluorescent dye coupled with specific antibodies is called fluorochromic. Third is: resolution is the extent to which detail in a magnified image is defined. Fourth is: fungal stain is a gram negative stain. Fifth is: acid-fast bacteria are resistant to decolorization. Now, this is a match the following. I have written left side 1, 2, 3, 4, 5, 6, 7, 8, 9. I'm sorry for the wrong now numerology. And on the right side, A, B, C, D, E, F, G, H, I. So on the left side, the different stains are written. One is gram stain, calcofluor white, direct smear, carbon fusion dye, crystal violet dye, kohler illumination, saffron dye, and gram iodine. And on the right side, you have to match. Hot method for acid fast stain. B is gram staining of clinical material. C is primary stain in the classic acid fast stain. Primary stain of the gram stain. E is focuses light path for proper resolution. F is counter stain in the gram stain. G is stain used to detect fungi in clinical material. H is principal stain used for microscopic bacteria examination. And I is mordant that forms the intracellular complex. So the students can answer, they can match from left side to right side, and they can write in the chat box the answers, so that whatever has been taught can be understood and can be answered accordingly. So we can understand whether today's class I could make you understand, or today's class is was of any benefit to the students or not. I think that is my last slide. Yeah. I have come to the end of my session.
Thank you, Dr. Sunita.
Thank you.
It was really very simplified and very interesting, and I'm sure the PGs have learned a lot, especially the first year PGs, because these are all very confusing concepts sometimes, and people get very stuck when they, study all these microscopes together. They get very confused. So I'm sure you can even watch the recording, all of you, on our YouTube channel of IAMM PG Masterclass. So, with that, I think there are no questions. I can't see any questions. Is there anything specific? Please let us know. So I think we can bring this class to an end if people don't have any question. Thank you once again on behalf
Thank
of
you.
IAMM and ICMM,
Thank
Dr.
you.
Sunita.
Thank you. Thank you so much,
Yeah,
ma'am.
somebody is asked interpreting. How do you interpret acridine orange?
Um, Interpretation of acridine orange, like
Yeah, even I didn't understand her question. She says, uh, interpreting acridine orange. Maybe Dr. Shreya, you can unmute yourself and ask the speaker yourself what you're trying to ask.
Good evening, ma'am.
yeah, please ask.
And how to interpret actinian orange means the microorganisms seen in the microscope, like the different colors are seen in the orange,
Yeah,
yellow. So how
yeah,
to differentiate the cells?
yeah. It takes up the acridine orange, the orange, and it appears orange to the eyes. So that is how we interpret it, and we find it positive. It is orange color. It is positive. That is
Okay,
how we
thank you.
interpret it. Yeah.
All right. Thank you. Thank you so much, ma'am. So with
Thank
that, we've come to an end. So next week on 6th, we will have a class on another interesting topic, and I'll soon be rolling out the flyer for that, or for 6th and for 20th of February. So do join us, everybody, then.
thank you.
Thank
Thank
you,
you.
ma'am.